current models and future possibilities. And before I actually get going, I thought I'd tell you a little bit about myself. So I'm actually British, so I'm from the UK, and I started my career as a student working with the Royal Botanic Gardens Kew. So Royal Botanic Gardens Kew, it's the big botanical gardens that is close to London, but they also have another site in Sussex, which is called Wakehurst Place, and this is the Wakehurst Place mansion. And that's where I started as a student, and actually in this old building, we had some laboratories, which was where we were doing our research. So I was an undergraduate student there, and then I did my PhD there as well. So it was at that time, it was the Seed Conservation Department of Kew. So it was a small seed bank, a small collection of wild plant species and seeds from the plants growing in the gardens. But by the time I finished my PhD, the seed bank had expanded considerably, and this was the start of, when I finished my PhD, was the start of the Millennium Seed Bank project. So this is the Millennium Seed Bank building. You can see here. If you were to visit Sussex, it would be a very good place to visit. It has a seed bank, which is actually underground, and then it has research laboratories and offices. One of the first things that I did after my PhD finished was to collect and do research on seeds from UK aquatic flora. So the Millennium Seed Bank project, it was about collecting seeds from 10% of the world flora from across the globe. But the first thing we did was actually to collect seeds from the whole of the UK flora. And there were about 70, 80 aquatic species in the UK. And I would travel around and collect these seeds. And we weren't sure if these seeds would be okay to be put into the seed bank storage. So my research then focused on seed storage behaviour of these aquatic species. In 2009, I left the UK and moved to the International Rice Research Institute in the Philippines. And I was working there for the rice gene bank. So this is one of the largest single crop gene banks in the world. When I was there, we always said there was about 110, 120,000 accessions, so different types of rice stored in the gene bank there. So you can see this is what we call the active collection, the medium-term storage collection. A couple of staff wrapped up in big coats because it's a bit cold in there, retrieving some samples, and that might have been for distribution, or it might have been for viability testing. You can see some of the team actually cleaning seeds, so a lot of that was done manually when I first got to IRRI. And then here, this is a drone picture of some of the regeneration plots. So every year, some of the accessions have to be regenerated to produce more seeds. And this is just a picture of that regeneration. And you can see the variety there in that picture, variety in the rice accessions. So I was in the Philippines for about 8.5 years, and then I moved to the Department of Agroecology of Aarhus University in Denmark. This is just a picture of Denmark, so just to get your orientation. Over here is Sweden, and down here is Germany. And Aarhus itself is here, but we're based down on the island, Zealand here, so closer to Copenhagen. This is our research building, and I think about 150 people work here. We don't have a lot of undergraduate students here, so we have some postgraduate students who do their research work here. And then this is our seed laboratory. So you can see some incubators there and various pieces of equipment. So that was just bit of background. The actual outline for my talk today, I'm going to start off by saying a little bit about why seed storage is important, how we measure seed longevity, and how we then model seed longevity and future possibilities. And I'll be mainly talking about this in the context of long-term storage, particularly in seed gene banks. But of course, seed storage is as old as agriculture. So as soon as crops started to be domesticated, the early farmers were collecting seeds to store and then plant the next year. And this is a traditional form of seed storage, so you can imagine that something like this might have existed many, many thousands of years ago. This is actually a picture that I took in Nepal, so it's a traditional form of seed storage in Nepal. I just want to also just give a definition of seed longevity, since that is what I'll be talking about in particular. And we can define it as the length of time before the viability, that's the ability for seeds to germinate. So before the viability of a seed lot reaches a critical level. And the critical level, it might depend on the context. We often talk about the time for viability to fall to 85% or to 50%. It could also be to practically 0%, so when there's no germination at all of a seed lot. And something else I wanted to say to start with is that seed longevity, it's really a trait that we talk about in the context of orthodox seeds. So we have two main types of seeds, recalcitrant seeds and orthodox seeds. I'll start with the orthodox seeds. So these are seeds that tolerate drying to low moisture content and then storage at low temperature. So usually we're talking less than 0 degrees C, and gene banks typically store at about -20 degrees C. Another important thing about orthodox seeds is not only do they tolerate the drying and the low temperature storage, but actually their longevity increases in a predictable way with decline in moisture content and temperature. That is within limits. I won't go into that so much today, but this increase in longevity with decline in moisture content and temperature, obviously it is something that we exploit. These seeds are usually air dry at dispersal, so they naturally dry on the plant unless they come in a fleshy fruit. And there may be some dormancy depending on the species, and it may be quite deep dormancy, maybe quite complex dormancy. And examples of orthodox seeds include most of our cultivated crops, cereals, legumes, temperate and arid land flora. At the other end, so we do talk about this being a continuum. So at the other end of the continuum, on this side, we have the seeds which are what we call recalcitrant. So these are desiccation intolerant. So the seeds, they die upon drying. So they don't tolerate loss of moisture. So of course that means that they're dispersed with a high moisture content, and they're often quite large. They're usually non-dormant at dispersal, so actually once they fall to the ground, they might germinate very, very quickly. And this includes species, so tropical rainforest tree and shrub species, some temperate tree species, and some aquatic species. So as I said, when I first started working for the Millennium Seed Bank, I was looking at the storage behavior of aquatic species, and we had the hypothesis that some of these aquatic species, because they were growing in water, dispersed into water, they might not actually have the ability to tolerate desiccation. So that's what I was exploring there. And it's true, some of them were recalcitrant, but actually quite a lot of them were orthodox. In the middle here, I've got two other sort of subcategories. Here we have minimally or suborthodox. So we do have some species which seeds are orthodox, so they do tolerate drying, but they're actually very short-lived in storage. So even though their longevity increases with decline in moisture content and temperature, we're still only getting them surviving a year or two at best, even under very good conditions. Sorry, I've got a message here. Can I please?
I think everything is good. Someone just asked, speak up, but we can hear you. It's okay.
Okay. So that's suborthodox, so very short-lived orthodox seeds. And then we also have another category, which species which produce seeds which might tolerate drying, but they don't tolerate low temperature storage. So these are what we call intermediate species, intermediate seeds. So why is seed longevity important? Firstly, it's important because it's about maintaining the viability, so the ability of the seeds to germinate, and the vigor of crop seeds, and that's from harvest until the seeds are sown. So this is very important from the perspective of seed companies and commercial seed lots. They want the seeds to have high quality when they're actually planted. Where I work the most is seed longevity in the context of conservation. So here it's important for the conservation of the gene pool, the genetic diversity of domesticated species and their wild relatives in gene banks. And here is a picture that I took at the ICRISAT gene bank in India. It's also important as a strategy for the long-term conservation of plant diversity in seed banks. So in seed banks, like the Millennium Seed Bank, which doesn't conserve crop species, but conserves wild species. So it's conserving the plant biodiversity. I'm going to start off by talking about this first one. The seed longevity is very much related to the vigor of a seed lot. And it's difficult basically to separate these two. But when we're talking about the vigor of a seed lot, often we're talking about things like the speed of germination and how well the seeds will establish when they're actually planted in the field. So what I'm showing you here are some germination progress curves. So the germination progress curves, they show you the cumulative total germination. So if you put some seeds to germinate, you'd follow the germination of all the seeds in that petri dish or however you sow them. And over time, you'd record the number of seeds germinating and add it all up so you get the cumulative total germination happening over time. And here are different germination progress curves, theoretical curves, seed lots which have been stored for different periods of time. So what you can see here is this first one here. So this is let's say a fresh seed lot, it's not been stored. And you can see it's germinating very quickly and it's reaching 100% germination. The longer the seeds are stored, the lower the final germination and the slower germination gets. So you can see that these germination progress curves are getting less and less steep, and the final germination that we end up with is getting lower and lower. So what we would say is that the vigor of these seeds is declining with storage period as the seeds age. We can calculate various parameters from this germination data, from this type of germination data. So examples include the mean germination time of all the seeds, time to 50% germination, so that would be here. So you go along here, and this would be the time to 50% germination. This is for this coloured one here. We might talk about the uniformity of germination. So for example, the length of time between 25 and 75% germination. So the time here, so that's about 28 days there, I think. Another measure is the area under the curve at a certain time point. So you can see I've got an area shaded in here. So that would be the area under the curve. after 32 days, and that's for this particular one. And you can see the area would be much greater for this black one, say, so a much greater area under the curve for that black line, where the seeds haven't aged so much. So all of these are examples of measures of seed lot vigor. What we can also see is that after a certain point in the germination test, the aged seed lots, for the aged seed lots, the seeds that do germinate after that particular time point cannot develop into normal seedlings. So again, for this line here, these ones would be the normal ones, and then these ones are all abnormal. This is that same graph here, and then I'm plotting it over on this side as what we call a seed survival curve. So at the storage period 0, you can see we've got the 100% germination. And then as the seeds are aged, germination is declining. And you can see how that's happening here, all these different time points. This is our seed survival curve. So that's the final germination plotted against storage period. This one here is the total germination. So it's the abnormals plus the normals. And this line here is showing you just the number of the percentage of abnormal seedlings of these total seeds that germinate. These are some pictures of what we mean by normal and abnormal seedlings. So these were taken by my PhD student here, Shabnam. So here she's doing experiments on some pea seeds. This is an example of a normal seedling, and you can see how healthy that looks with good roots and growth. Whereas here on the right, we have some abnormal seedlings, and you can see how they're very stunted, and the actual seeds themselves are looking very shriveled here. As I said, this is a seed survival curve, and in this case, it's for the total germination, so normal and abnormal germination. What we see when we do these types of experiments is that the decline in vigor occurs before there's an obvious decline in viability. So what you can see here is the viability of the seeds being maintained quite well for some period of time. So we're still getting 99, 100% germination. This line here is showing you the vigor of the seed lots, so the speed of germination and so on. In this particular case, it's the area of the curve of a germination progress curve. And you can see that that's declining immediately. So as soon as the seeds are aging, we're getting declines in the vigor of the seeds. Of course, this is a big concern for seed companies. You can also see that in this picture here. So this is some pictures of seeds from a storage experiment where the seeds have been aged for different lengths of time. So this is 1 day, 20 days, and 30 days under these particular conditions. They were all sown to germinate on the same day. And you can see how the vigor of the seeds is declining. So here we've got all the seeds, most of the seeds germinating well. They're looking very healthy. You can see here at the same time point during the germination test that although some of these seeds will probably develop into normal seedlings, they're a long way behind these ones here. And then here you can see how the viability has declined a lot as well as the vigor. So if any of these seeds did develop into a normal seedling, it's going to be very, very slow and for a long time after these ones have. So the vigor starts to decline immediately. So very early on during storage. The potential longevity also starts to decline immediately. And I've plotted that here as the column graph. So this is the potential longevity of the seeds. And again, you can see just how it's declining with any aging of the seeds and going down quite dramatically. I've also plotted that out here. So this is our original seed lot. And then if we took the seed lot here, this red one, if we took that and put that into storage, you can see it's losing viability before this black line here, before this black seed lot with no aging. And again, with the blue one, again, you can see the curve is going down earlier, and so on and so on and so on. So the potential longevity of the seeds declines as the seeds age. As I said, we often measure seed longevity as P50, the time when viability falls to 50%. So you can see how longevity of these seed lots is declining as the age of the seeds, as the physiological age of the seeds increases. So that's a bit about how seed longevity and vigor are related. I then wanted to say something about how we actually measure seed longevity, and I've actually alluded to this already. So here's the definition again. Seed longevity is the length of time before viability of a seed lot reaches a critical level. We usually determine it under very controlled conditions when we're doing what we call seed storage experiments. And what we mean by controlled conditions is that we control the moisture content, and we control the temperature. And there are two ways, main ways of doing this. So first of all, one way is to get your seeds and adjust the moisture content, and then put the seeds in airtight pouches so that the moisture content is then fixed, and then put the pouches at an aging temperature. So this is an example of that here, where the seeds are placed in a particular RH environment, so they're over a solution, creates a particular relative humidity. So the seeds equilibrate to that relative humidity, so they take up moisture, and then they're packed in aluminium foil pouches in this case, and they're put in the aging oven. Another way that it can be done is to have them in this setup here, and this setup would be put at the aging temperature, so that's the controlled humidity and temperature in that way. We're usually raising the moisture content of the seeds because the seeds that we have, that you buy or whatever, are usually dry, and we want to accelerate the aging process so that we get results within a reasonable experimental time frame. So we usually have to raise the moisture content and raise the temperature. And depending on the species, we might be talking about having seed moisture content about 10%, 11% relative humidity, moisture content, equivalent to about 60% relative humidity. And we typically do the aging at 45 degrees C. But we're also interested in longevity in other environments, in particular in seed gene bank conditions, but also in the soil seed bank. Seed gene bank conditions, so these are when seeds are usually dried to about between 5–7% moisture content, and then stored either at 2 to 4 degrees C or at -20 degrees C. Soil seed bank is a totally different environment, and I tend not to call this seed longevity, but seed persistence. So the seeds, obviously they're dispersed into the soil, and those conditions in the soil will be highly variable in terms of both moisture content and temperature. But obviously it's particularly important in terms of understanding the regeneration of vegetation, but also if you're thinking about weeds, you're wanting to know how long the weed seed bank is going to be viable for and cause a problem for the crops that you're growing on that particular plot. But if we're doing a seed storage experiment, we have the seeds in this very controlled environment, and then we'll take samples out at regular intervals to do a germination test. And when we're scoring for germination, we might be talking about the total germination. We might talk just about the normal germination, or even just waiting for the seeds to show radical emergence. So we might score at a very early stage. So this is the type of data that we would get. So this is plotting germination against storage period, as you've already seen. And the data is most often modeled using what we call probit analysis. And this is a generalized linear model fitting process that assumes that the variable, in this case the time to seed death, follows a normal distribution. So what you can see here, so this is the data that we have. And what you can see is that early on during storage, many seeds die between each time point. So you can see here to here, and that's shown here. Then the number of seeds dying increases, and you can see that here. So more and more seeds die between each of those sampling times. And then it slows down again. You can see that here, this slowing down, until eventually just one or two seeds are alive and they slowly die. This here is the normal distribution, and our probit analysis is basically fitting normal distribution to this data here in this format. So here you can see the results of what we call probit analysis. So that's the results here. And here the equivalent result in terms of fitting that normal distribution of seed deaths over time. So this is a probability density function. So you can see this is the equation explaining this curve here. And there are two main parameters to take note of for this probability density function. One is the mean here, so that's this. And this is actually the equivalent of our P50. This is the time when viability reaches 50%. That's the mean of the normal distribution. And then we also have a value here which is called sigma. And this value sigma, it gives you an idea of the spread of the distribution. So whether this L-shaped curve is very narrow or whether it's very flattened and spread out. And this equation here is actually the equation describing this line. So again, you can see we have the sigma reflected in here. This symbol here, this is telling us that we're doing the normal conversion. So we're converting the percentage value to what we call a probit value. And then this is the equation that we're interested in. So Ki is the theoretical initial viability in probits, and P is the storage period, and then, as I said, we've got the sigma here. Then the longevity of the seed lot depends on the actual storage environment. So here again, we've got some survival curves. And these survival curves might be for seed lots of the same species stored at different moisture contents or stored at different temperatures. And basically the seed longevity, so the survival curve, is spread out as seed moisture content is reduced or temperature is reduced. So the seed longevity increases as we reduce the moisture content or the temperature. And of course, this is one of the things that I said was part of the definition of orthodox seeds. You can see the respective probability density functions of these survival curves. So you can see how the mean, the P50, is getting shifted, so the longevity is increasing. And also how the sigma value is increasing. So here the sigma value is very small because it's a narrow distribution. And here it's getting bigger and bigger as the normal distribution gets flattened and spread out. So if we took value of this sigma and plotted that then against moisture content or temperature, we'd get relations that look like this. And we can sort of say that this sigma value here for these types of plots are giving an indication of the longevity of the seeds. So this y-axis you can think of as longevity, and you can see how the longevity is increasing as we reduce the moisture content or as we reduce the temperature. And the effect is quite considerable when we talk about moisture content, less so for temperature. This is actually just over 10 degrees C, whereas this is quite a big drop in terms of moisture content. Of course, we can fit relations to these two plots. And these are actually just linear axes now, but if I make them log axes, so this is now a log scale here and here. This is a log scale, but this is a linear scale. You can see that we get straight lines. And so then we can end up with a full equation describing how describing the viability of a seed lot depending on the moisture content and temperature that the seeds are stored at. And this is the viability equations of Ellis and Roberts, that are very well known and used in seed science. And so here we've got the Ki, which is that initial viability that I mentioned before in probits. Here we've got the storage period. And then in this bit, you can see here, this is where we've got the moisture content, and here is where we've got the temperature. So these bits are describing these two parts of the relations here. And we can then use this equation to predict the longevity of any seed lot of a species where we've solved this equation. And we can use it to get predictions of longevity under any storage conditions within those limits that I mentioned, but there are some limits to where this equation is actually applicable. It does apply over most of the storage conditions that we tend to use when we're wanting to conserve the viability and vigor of seeds. In the literature, there are other ways of analysing the data from seed storage experiments. And so one that's been used in particular by Christina Walters is something that's called Avrami kinetics. So this is an equation developed for explaining the rate of chemical reactions. And she applied it to germination data, so that's then how it looks like here. And you can see it's fitting the data quite well. It doesn't take into account the statistics of testing seeds. So actually, when we're talking about germination, each seed can either germinate or not germinate, and that means that we end up with what we call a binomial variable that we're analysing. And the probit analysis takes that into account. So it takes into account the likelihood of each seed germinate, giving the viability of a seed lot, whereas this particular equation doesn't. And there are other ways of analysing data, and these are ways that have been applied perhaps in particular to gene bank data, because there are quite a lot of issues with analysing germination data from gene bank storage. So different ways have been used. And this is an example from a paper, a group in Japan. So they looked at the historical viability data for seeds, different species stored in their gene bank. These are the conditions that the seeds are stored at there. And what you can see here is instead of looking at the actual germination of a seed lot during storage, they looked at the overall performance, if you like, of all the seed lots of a particular species during storage, and the probability of maintaining, in this case, viability above 85% or more of the initial germination. So they're not looking at actual germination, they're looking at the seed lot, whether or not a seed lot loses viability below 85%. And then they use something that's called a time to event analysis, and so they actually got out what's called Kaplan-Meier estimates, but it's just a completely different way of looking at the data. We did something a little bit similar here. So this is data that we looked at IRRI. And again, we looked at the proportion of accessions that were generated in particular crop years, proportion of accessions not failing, the ones that still had 85% viability. Whereas these ones here are the ones that did fail, and these are the seed lots that had less than 85% viability. And this is just split depending on whether the seeds were stored in the active and base collection. And you can see these are the different storage conditions for those two different collections. So we expect the seeds in the base collection to stay viable for longer because they're stored at a lower temperature. In this particular case, we were wanting to understand the longevity of the seeds. And as you can see here, so these seed lots here, so these were the oldest seed lots. And there was a decline in the viability. So these seeds have been stored the longest. And when we looked at the data, their viability, and you can see that they are showing a higher rate of failure, if you like. But we also saw that over on this side. So even though these seed lots were much younger, so they hadn't been stored for so long, these seeds were also showing a greater rate of failure. So these overall, all the accessions were produced in later years. So this was actually very concerning for us. It suggests a decline in the quality of seeds that are put into storage in later years. And one of the things we were trying to do is to actually understand why that was. So that's already talking a little bit about gene banks, and there were a couple of examples of how different groups have analysed historical viability data from gene bank storage. So then I thought I'd just say a bit more about the role of gene banks, in particular in the context of food security. So a seed gene bank, and I've put seed there because there are different types of gene banks. But in the case of a seed gene bank, we're storing a sample of seeds, and that's usually called an accession, that represent the variety, landrace, or wild relative of a cultivated crop. Gene banks, they build up their collections from making collecting trips. So this might be collecting trips to wild areas to collect perhaps a wild relative. It might be going around to farmers and asking farmers what they're putting in the field and asking to have some. There might be donations from farmers or from breeders or from other scientists. So this is how many of the gene banks assemble their collections. Seed gene banks, the use of the material is usually for research and breeding related to food and agriculture, because we're talking about crops. And legal instruments, so the legal framework in which they work, and by work I mean by which they acquire germplasm and then distribute germplasm, they are the CBD and Nagoya Protocol, the International Treaty on Plant Genetic Resources for Food and Agriculture, and that's for a particular group of species, of the most important crop species, and then local country access and benefit. Key activities of a gene bank are the acquisition of material, so as I said, from sources, so that's called acquisition. It is the storage of the seeds, and that's long and medium term storage. It will be viability monitoring, and that's why we end up with viability data that we can actually analyze for longevity. Characterization, so this is scoring the plants, different parts of the plants, for particular agronomic traits which are thought to be highly heritable. And then this characterization data is made available to users so they can select material that they want to try in their experiments or breeding or whatever. Distribution, so sending material out, samples out to users, and this is a very core function, a very big activity at some gene banks. And then multiplication and rejuvenation. So this is producing more seeds either due to loss of viability or because the number of seeds in storage has declined. And the reason for that decline is because the seeds have been distributed or used for viability testing. And here are some pictures from Norgene, which is the Nordic Gene Bank, which is in Sweden, so not so far away from us. So they have a lot of freezers. You can see inside here boxes with the seeds in. And this is an example of how they do some of their regeneration. So you can see they've got isolation cells here so that there's no cross-pollination and they keep the accessions separate. As I said, the role of the gene banks is very important for food security, and they are specifically mentioned in the Sustainable Development Goals. So this Sustainable Development Goal 2 is about zero hunger, and target 2.4 is maintain the genetic diversity of seeds, cultivated plants, and farmed and domesticated animals and their related wild species through soundly managed and diversified seed and plant banks. That's where the gene banks fit into helping towards food security. This is just an overview of the gene bank operations. So starting with incoming germplasm, so this is the acquisition of new materials. So this is where I said there might be collecting or there might be donated to the gene bank. Often a sample of that original material would be put into what's called a seed file. So this is a reference sample so that later on you can evaluate to make sure that the seeds in storage match the original sample that was donated, so that match the seed file. The amount of material that usually arrives at a gene bank is usually quite small. So usually the first thing that does have to happen is seed multiplication. So the seeds will be planted out. It might be necessary to do that under quarantine if it's material that's coming from a different country. So the seeds are multiplied, they're harvested, cleaned, dried, sorted, and then they're packed. And at this stage, there'll also be samples taken for phytosanitary testing, so checking for seed health, not carrying any diseases, which would mean that the seeds couldn't be distributed. And initial viability tests, so make sure that the quality of the seeds, the viability of the seeds is high enough. And then the seeds are packed for storage. And here we've got seeds put into long and medium term storage. So this one is the active collection, this is the base collection. Samples are also prepared for what we call safety duplication. This is where one or more samples are sent to a gene bank in a different location as a backup. So in case something happens at the main gene bank, it will be possible to retrieve a sample and then regenerate that material. So most gene banks now send duplicate samples to Svalbard, the Global Seed Vault, Svalbard. And the ICARDA Gene Bank, I don't know if you've heard, but the ICARDA Gene Bank, when ICARDA, which is an institution, when they had to leave Syria, so they were based in Syria and they had to leave, and basically then they had to restart the gene bank from scratch. So they actually requested the samples that they had sent to Svalbard, they requested to have them back. And then they've been basically rebuilding up their gene banks, so producing seeds from these safety duplicate samples to get the gene bank operational again. The seeds in medium-term storage, so this is 2 to 4 degrees C. So as I said, this is the active collection, so that means is it's used for distribution. Whereas the long-term storage, they're at -20 degrees C, and that's long-term conservation. But they will all be used for viability testing. So viability tests will be carried out regularly. And then when either the quantity declines or the viability declines, this cycle will start again. So the material will be regenerated, cleaned, dried, sorted, and packed and put back into storage. Here I've also got characterization. So as I said, all the accessions will be characterized for highly heritable morpho-agronomic traits. And that's part of the data that's made available to users when they want to select GeneBank material. So data is very, very important as part of GeneBank operations. So the collection and management of that data, it happens at every stage. So here with incoming germplasm, this is called passport data, all about the material that's already donated. But then all the operations data is also collected there, and it's also driving the operations as well. So it's a result from a viability test that then goes into the system, and then it triggers the need for regeneration. So modelling GeneBank viability data. So it is difficult, and in a moment I'll show you another slide with some graphs which will show you just how difficult it is. But at the moment, what we tend to do with the viability testing, sorry, if I just go back here, is we tend to do this viability testing at standard intervals. So typically every 5 years for seeds in the medium-term and storage, and every 10 years for seeds in the long-term storage. But if we were able to improve our ability to model viability data, so model the longevity of the seeds in GeneBank storage, it would make GeneBanks more efficient. So in theory, we might use fewer seeds, less staff time, and other resources for viability testing. It would give us better prediction of our viability testing needs and also our regeneration needs. So how often we need to test the viability and how often we might need to regenerate a particular accession. So we want to do something better. We have a saying in English, which is, if it ain't broke, don't fix it. So what that means is basically don't start all over again. And when I'm thinking about how we might improve how we model seed longevity, particularly in relation to GeneBank viability data, I don't think we need to start from scratch, because we already have a good model which describes the longevity of seeds. So that's the viability equations. And as part of that, as I said, it basically assumes that there's a normal distribution of seed deaths over time. And this does make biological sense, because we're talking about a population of seeds. So it's quite a sensible idea that that would follow a normal distribution. And then compared with some of those other ways that I showed you of where people have tried different things for modelling the data from GeneBanks, the viability equations do give us some very nice parameters which we then know we can apply in terms of modelling the effect of moisture content and temperature on the longevity of seeds. The big problem that we have is that we still have very limited data from GeneBank storage where we can actually apply the viability equation. And that's actually because many of the GeneBanks have a viability threshold of 85%, so we don't get enough data when the viability is actually declining to get a good fix on the PROBIT model when we have that data. The other problem that we have is that the data don't always conform to the assumptions of the viability equations. And one in particular, so the viability equations, there's the assumption that all seed lots within a particular species will lose viability at the same rate when they're stored under the same conditions. So all the rice seed lots in the IRRI GeneBank, we expect to lose viability at the same rate. But actually, that's what the viability equations would tell us. But in fact, what we think is that there's quite a lot of variability, and we're only just starting to understand how much variability there actually is. So here's some example of some GeneBank data. So this is from a paper that we did last year, where we analysed historical viability data from a number of different gene banks. On the left here, it's data from the Gene Bank of the International Institute for Tropical Agriculture in Nigeria. And so 2 Vigda species. And what you can see here, so this is their real data, and what you can see is that basically they had anywhere between 0 and 100% germination, regardless of the storage period. So there's no obvious pattern in the data. It was just all everywhere. And we think part of this was just due to dormancy, so that they were doing tests and weren't overcoming the dormancy, and this is why we were getting these results here. So not really a true reflection of the viability of the seeds. And over on the right here, this is data for some rice seedlots from Africa Rice. And this is again a pattern that we've seen from different gene banks where dormancy isn't such an issue. But here, basically, we're plotting germination against storage period, but we've sorted it between 2 observations. So we've got the initial and then the most recent. And we've sorted it according to the result of the initial viability tests. So here are seedlots which had an initial viability of 100%. And you can see then that the subsequent test result, it could have been anything. So whereas we might expect it to often be high, there are quite a lot which are showing quite a low viability. If I go over here, this is where the initial viability was 85%. And then the subsequent test, you can see we have, where we've got data, you can see for some of them it increased and for some it went down by different degrees. This could be because of that variation that we're starting to understand in the longevity of seedlots, even under the same storage conditions, or it could actually just be due to random sampling error. But it does mean it's very difficult to get any idea really of the longevity of seeds in gene bank storage. We're still doing quite a lot of work. Really what we're thinking is something like this. So we're going to take different things that we know and put them all together to come out with better customized predictions of seedlot longevity. So here we've got the routine viability monitoring. So that's what we're doing already. On top of that, we would also put in historical viability data. So we will get an understanding of how the seedlots of the same species or variety group or accession have performed in the past. We will also use the viability equations where we can to get predictions of longevity. And then what we'll also do is to do seed storage experiments at the start of storage. So instead of putting all the seeds into gene bank storage, we'll take another sample and do an actual seed storage experiment. And this will give us both the initial seedlot viability, also give us an understanding of that variation in the rate of loss of viability and a ranking of seedlot longevity. So hopefully by putting these all together and perhaps into an AI model, we can come up with these better predictions of longevity. That's what at the moment, we're mainly relying just on this. So this would be building these things in. But the other thing that's going on in quite a lot of seed research is that people are trying to understand what's happening inside the seeds as they age, to see if we can come up with a different type of marker of the age of the seeds and the viability. So quite a lot of research is going on here. So it might be that we don't need to do an actual viability test, but we rely on this marker. Whether that would be with or without any of these other things, I don't know. So that's just where I wanted to finish. So just nearly on an hour, I think. These are just some pictures, some other pictures of gene bank operations. So this is a picture of the drying room at Africa Rice. So this is at about 15 degrees C and 15% RH. It's where the seeds are dried and then packed for storage. It's also where they take samples out for distribution or for viability testing. Here we've got some regeneration plots. So this is at Ikada in Lebanon. You can see that they're putting bags over the plants, and that's so that they don't lose any of the seeds. So otherwise the seeds would get dispersed, and then they wouldn't have enough seeds to put into storage. This is some viability testing of bean seeds at CIAT. This is in Colombia, and you can see they do their bean seed testing in sand here. And then this is also CIAT, but this is their old gene bank. So this is just another picture of gene bank storage conditions. And with that, I'll say thank you very much. Hope you've managed to follow. I'm happy to answer questions, but also if you'd like to get in touch, here's my email address.
Thanks,
Thank you.
bunch, Professor. I appreciate. Can you hear me?
Yes, I can.
Good. I appreciate on the behalf of all the participants and members of the Agronomy Scientific Club, Tehran University. Thanks a bunch. I'm going to open up the chat box so the participants can write the questions, or even you can raise your hands.
So there's a very good question there already from Babak. Is that how you say your name?
Yes.
Sorry if it's wrong. So explain more about the factors which we have to consider while predicting seed longevity of wild species. So often with wild species, we don't actually have much information to know their seed longevity. And really that's, you know, if you're starting with a new species, we really don't know very much. We did some work when I was at Kew where we basically looked at ecological traits and to see how they might predict longevity. And basically what we found was that seeds from cooler, drier environments tend to be longer lived than those from warmer and wetter habitats. And I guess because I guess you're probably talking about species which are from Iran and then probably from quite dry area. Correct me if I'm wrong. So you might predict them to be quite long-lived, but really we don't have any idea when we're starting a new species from scratch. And the only way to understand then would be to do a storage experiment. And we've done quite a lot of, and this is what went into that work. And we do quite a lot of what we call comparative longevity experiments to try and understand, you know, this variation in longevity across species. In terms of dormancy, when we're looking at longevity, we like to take out the issue of dormancy. So we're just looking at when we're doing the germination test, we're just looking at the viability of the seeds, and we don't have to worry about dormancy, if you like. For some species where there's a physiological dormancy, you'll actually see the dormancy being broken during storage. So instead of it just being a curve going down, it'll actually you'll actually see it increasing at the start of storage and then going down. So you'll see the breaking of dormancy and then loss of viability. If it's something like physical dormancy, then actually if you're wanting to do storage experiments, one of the most important things is actually because you want to raise the moisture content, it's important to scarify the seeds before you start the test so that the seeds can equilibrate to the environment and take up moisture, and then when you pack them and put them in storage, you know, they'll be at the target moisture content that you want. But it's not so easy. So that was a good question. Is there another question coming up?
I think someone is typing. All the participants can raise their hand to speak up. Ms. Zamani, you are a host, you can speak.
Okay, thank you. Let me start in my base. Hello, Dr. Hay.
Hello.
Hello. We have a honor to have you for this meeting, and I'm really glad to see you online. I have a question. As you are one of the member of ISTA, the International Seed Association testing, I have a question. We have many species in the world which don't come in ISTA on notebook. You know, about the cardinal temperature, storage condition, and key features. Are there any condition we have introduced some species to this notebook? We add some species to this notebook, and what is the solution? What is the way for introducing? Because I'm a graduate student in MS, Seed Science and Technology at the University of Tehran, and I worked 2 years on seed Hamada salicornica, but we don't have any information in the ISTA notebook, and I wanted to introduce something about this. What is the solution? What is the way? Thank you so much.
Yeah, so the ISTA rules, they do cover quite a lot of species, but they are mainly species which are traded, you know, so they're important commercially. So then, of course, there's, you know, thousands of species which aren't covered at all. And there will be, I mean, they are mainly interested in having rules for these commercial species. So if you have a species which you are interested in but doesn't really have any commercial value, it might be hard for you to get those into the ISTA rules. If it's something that does have significant commercial value, then you'd basically have to you'd have to contact either ISTA or the technical committee for the particular test that you're interested in having in the ISTA rules. So if that's the germination test, you'd contact the germination committee. If it's the moisture content test, you'd contact the moisture content committee, and so on. And then, you know, you'd have to persuade them that it was important enough that they would then do a validation test. So there has to be preliminary work to start with. So in the case of germination, you might have an idea which is the best conditions to germinate your seeds. But they would then have to check that and compare it to some different conditions, but then also make sure that the results are reproducible across different laboratories. So it's quite a long process to get a new species covered by the rules. But yeah, I think, you know, it won't happen for a lot of species. But if you have a particularly important commercial species, then you could propose it and try and get things moving.
Thank you so much.
There's a question in the chat from Hamid Reza. Are there any specific software for predicting seed longevity? If you've got time, I can just share my screen for a moment. Let me. I'm just going to try and go to a website which I can show you.
You can share your screen if you don't mind.
Yeah. So hopefully you can see my screen. Yes?
It's clear, thank you.
Yeah. So this website, so it's called the Seed Information Database. So it's hosted, it was created by Kew. So this is the website. You can also just search Seed Information Database. You should come up with it. You could put in Kew as well if you wanted. They've been saying that they're going to take it down for quite some time. So you can see it was supposed to stop working at the beginning of May. It is still working at the moment. And what I can tell you is that I don't know how long it's going to be going for, but there is another initiative with a group of researchers in Australia, part of the Society for Ecological Restoration. So they're sort of taken all the data from the Seed Information Database here, and they're trying to make it available for when it's not available here at Kew. And hopefully when that happens, Kew would actually send a link so that you would know where to go. Seed Information Database, it's great. It has a lot of information on thousands and thousands of different species. So for example, if you have a species and you don't know how to germinate it, you could put it in and see if there's any germination data for that species. In the first instance, it's populated with information that Kew itself has collected as part of the Millennium Seed Bank work. Over here on this sidebar, you'll see a link here to seed viability constants. If you go here, you get to this page, and this is where you can basically implement seed viability equations to get predictions of seed longevity. So let's say, let's do this one, predict final viability. So as I said, with the seed viability equations, they do have to be solved. So what that means is that the parameters of the equation that describe the effect of moisture content and temperature on longevity have to have been determined. We have a bit of a shortcut in that we expect the response to temperature to be the same for all orthodox seeds. So then it just has to be solved for the moisture content. But these are the list of species where the viability constants of the viability equations are available. And we choose, let's just try a chickpea. You can see these are the values here. So these two describe the effect of moisture. This one here is the main one about the effect of moisture content on longevity, and these are the temperature ones. And then we put in the moisture content of the seeds, and you can either put in a value, just whatever you like, or you can get it to calculate a value. So let's say the particular chickpea that we're looking at, we know it has an oil content of 5%. Let's say we've got them at about 30 degrees C and maybe about 20% relative humidity. It'll then predict the moisture content of the seeds. And this is actually based on a different equation, which is called Cromarty's equation. That's 6.2%. And if we put that in here, 6.2, and let's say we're storing at 25. Let's say our initial viability is 95, and we're storing, let's say, I don't know, 4 years. So you can see viability there that's predicted is 93%. So this is where you can get some predictions of longevity, and you can just go in and you can play around looking at different species, but also looking at different moisture contents, temperatures, and the effect of initial viability. So if I change this here, for example, just to 85%, so now you can see it's gone to 80%. So you can see how the effect of initial viability increases the longevity, impacts the longevity of the seeds. Okay, so that's the seed information database. Qassem, sorry if I've got your name wrong. Yes, you're right. You know, when we're talking about commercial seed lots, germination speed and vigor are important. And probably the best thing, where I can best direct you is to a paper by Kent Bradford. So Kent Bradford has done a lot of modeling of the germination rate, so those cumulative germination progress curves. He's done a lot of modeling of that process when seeds have different stresses. So it's all called threshold modeling, and it's, you know, he's done it for if you put seeds with PEG solutions, so they experience drought, and you know, modeling all of that process. But there's one particular paper where he modeled the speed of germination as the seeds aged, and actually those graphs that I showed you at the beginning were based on some of his modeling there. So it is basically possible to combine longevity and vigor. It's just that we haven't done it so much, but it is there, and it can be done. So it's a paper by Kent Bradford. I can't remember who the co-author was of the year, but if you want to write to me, if you can't find it, I'll send it to you. Babak, you've got another question. What is your idea for predicting seed longevity when we have fluctuating temperature or humidity? I know there are a couple of papers on this. So that there's one by, I think it was Hung Hong and Alice, from a few years ago. Again, if you write to me afterwards, I can send you the papers. And I think for that one, they just, was it temperature? I think they were moving the seed to different temperatures. But basically, what we think, and partly based on their work, is that there's no effect of the fluctuation. It's just the effect of, you know, the length of time at one temperature and then another temperature, or it would be the length of time at one moisture content and then another moisture content. But there's no interaction effect, if you like. So the fact of moving, it actually comes up, this question comes up in relation to gene bank storage, and whether taking samples out of the gene bank and then putting them back in again, whether that's actually having an impact. And we don't think so. We just think that the length of time when they're at the higher temperature, you know, obviously they're going to be aging a bit faster than when they're at the lower temperature, but there's no effect of actually moving them between the two. But we haven't done, I mean, I myself haven't done a lot of that type of work. I think there's only a couple of papers at the moment where they have, but basically we don't think there's an effect of the fluctuation itself. Have we finished with questions?
still cannot connect my webcam. It's not my day, I think. Okay, there's no more questions. I think we're done.
Is there any paper on finding dynamic formula for seed longevity? I'm not sure what you're getting at there. As I said, if you write to me, I'll just put my email address in again, so I can send you some papers.
I think it's done. I truly appreciate you and your time you spent helping and presenting us. We enjoyed every minute of that. Thanks a bunch, Professor, and we wish you luck and happiness.
Thank you. Thank you for inviting me again. Yeah, just get in touch if you've got any further questions, and good luck with your own research.
Thank you. Have a nice time.
And you, bye.
Bye.